Bonjour à tous. D'abord, je tiens à excuser l'absence.
Good afternoon. First of all, Professor Charpentier told me to tell you that she's very sorry. Unfortunately, she could not join us here tonight. So I will try and cover her work as well. And I convey to you her regards, of course. We will talk about protein yeast extract. We have 4 parts: a history of the project, then selection of the yeast and selection work on the yeast in order to obtain the protein extract. Then rapidly we will address industrial production and we will focus on feedback. As far as the history of the project is concerned, 3 assumptions basically are at the very basis of the project. First of all, the controversial aspect of animal proteins between 1996 and 2000 due to Charcot-Creutzfeldt disease. That entailed that the bovine gelatins had to be replaced. And then we have the aspect of allergens, casein, albumin, and lysozyme. And the third element is the willingness on the part of a number of countries, Nordic countries, to be strict about animal proteins. And I'm referring to swine, bovine products, but also fish gels. Due to that reason, we sought to find an alternative to animal products, and yeast was picked as a source of proteins. The project started several years ago, thanks to a cooperation between Sofralab and the IUVV in Gien, with the team of Professor Feuillat at the time and Claudine Charpentier. This team has been working for some time now on yeast derivatives, and specifically finding on lees and usefulness of yeast derivatives during the autolysis of yeasts on lees. In the course and during this work, Professor Feuillat had validated the usage of a partial yeast lysate that had proven to have a number of interesting characteristics for winemaking. This product, in specifically very tannic red wines, had fining properties. So I'm talking about 2 aspects in fining: clarification of wine and the improvement of the organoleptic profile of wine itself. If we consider the product closer, in order to understand why fining was occurring in some instances and not in other cases, we found proteins to be the responsible. They have a 14 kilodaltons molecular weight or less. That was detected by electrophoresis, and you see a model here. On the left-hand side, we have the track on the autolysated product developed by the professor, and here we have 1 strip less than 14 kilodaltons. Since these are low molecular weight proteins, we need special conditions in wine, that is, a charge of tannins and polyphenols, which are sufficient to account for the clarification of the product itself. Here you see a number of results of the tests made with this first generation of product, where you have the impact of the turbidity in function of the dose from 10 to 30 grams per hectoliter. And we have the effect of clarification going from 26 NTU to 9 NTUs at the dose of 30 grams per hectoliter. There is also an impact in terms of organoleptic features and characteristics. Initially, people talked about the product being dry in the mouth and a hard nose, a closed nose, while with this product, it was possible to improve the aromas both to the nose and in terms of the other organoleptic characteristics. And the impact was also good in terms of tannins. The wine, the product was less dry. The product, therefore, was interesting, and yet it was a first-generation product, while what we needed was a product that would make it possible to work with all wines and not only with very tannin-rich ones. Hence, the idea of using non-degraded native proteins of yeast in order to create a product for fining purposes. We performed a number of tests. To that end, in some cases, Professor Charpentier participated in the trials. The intent was that of obtaining a strain of yeast where lysis could be obtained with an incubation at 37 degrees. Why 37 degrees? Because that provides a proper lysis, and the intracell content in terms of protein remains sufficient, so that we still have proteins in their native form, that is, exceeding 15,000 Daltons. Several approaches were available. We studied literature in order to see what was available as knowledge. A first approach pertained to genetic manipulations. Articles had been published to evidence the usefulness of suppressing a number of genes in the metabolism of yeast with a programmed autolysis at 37 degrees. So things had been published about that by Professor Cebollero, for instance, and also by Professors Alvarez and Moliero. They described genes which were necessary and capable of causing a lysis of yeast at a certain programmed temperature. Another approach was that of direct mutagenesis. In this case, or directed mutagenesis. In this case, UVs are used as mutagenic agents. At the time, a number of publications were made on that, including the publication by Gonzalez, who had evidenced the usage of UV radiation to have yeasts rich in proteins and manoproteins. Our approach entailed going for mutagenesis, because the part of controlled mutagenesis using GMOs is interesting, because in that case you target the genes you want to have an impact on, and this can be very precise in obtaining mutants, but at the same time you have a disadvantage, because consumers in general oppose the usage of GMOs in food. And therefore, we went for directed mutagenesis. Of course, we also have a negative element there, because when you have this approach, you do not have a control on the type of mutation that you generate, and that entails a selection of yeasts which underwent the mutagenesis in order to get to what is of interest. After ascertaining the quantity of radiation that was needed, we sought to simplify methods in order to validate that mutagenesis, and that entailed picking yeasts which could be interesting and useful for our purpose. We went for screening techniques, simple ones, which made it possible to perform analysis on a pool of yeasts. At the time, we had, let's say, 100 potential candidates. And out of some hundreds, actually, of potential candidates, it's not that you can sequence all the genome, the entire genome of the yeast to identify the place of the mutation. Progress, of course, has been made in genetic testing, and yet it is still an enormous amount of work. And this also entails that you have knowledge about sequencing. You have to be able to work on the analysis of data. So we abandoned that option and we went for colorimetric methods. We started with them from the strain that we had that was very rich in proteins, and we applied a UV dose that had been selected in such a way as to be sure that the dose would give mutations in general without having an excessive exposure, so that there wouldn't be a total destruction of microorganisms. And we saw that at this dose there was a survival rate of yeast between 2 and 10% with that dose of UV radiation. We also assessed vitality with the colony system with erythrosin B. You inoculate clones on a petri dish with a certain amount of erythrosin B, and you then apply two temperatures, 24 hours at 20 degrees Celsius, and then an additional 24 hours at 37 degrees Celsius. This makes it possible, by using a coloring agent, to establish which yeast can be thermalized at 37 degrees. So when we have colonies that are dark red, that means that we have no viable element. And after the thermal shock, therefore, we have yeasts which undergo autolysis, and they are the so-called positive witnesses, while when we have still viability, you have pink as a color, where we have things that are less sensitive to thermal shocks. Thanks to this method, we can have a screening of the colonies of yeasts after the mutation in terms of the 37 degrees threshold.
There's another methodology which consists in the selection of thermally sensitive mutants using nystatin. After the genesis, the mutants were incubated at a temperature of 37 degrees C, then exposing them to nystatin. Those that could reproduce and that would be sensitive to nystatin, and therefore they will reproduce. Those that don't will be resistant to that antifungal agent. So that is an additional screening element that will make it possible for us to screen potentially interesting yeasts that are capable of doing autolysis at 37 degrees C. Nystatin is bounded to ergosterol in yeast and modifies the permeability of the cytoplasmatic membrane, causing the death of the yeast itself. Now, what was the reason why we used different selection methods? Looking at the temperature, each of these selection methodologies that I have just described, using nystamicin or erythromycin B, each of these methodologies has pros and cons to it. we wanted to use a maximum of elements so that we would be able to avoid to work in overly restrictive conditions, because we're working in a blind condition in order to have a sufficient number of yeasts to see whether they met the objective or not. Then another objective was described by Gonzalez, the methodology using colorimetry, which makes it possible to identify using a dye, in this case BCB, 5-bromo-4-chloro-3-indolyl phosphate, and it's a substrate which, in the presence of alkaline phosphatase, which is an enzyme in the yeast, it takes on a green color and makes it possible to quickly identify colonies that will be in an autolytic state. In the third stage, we went through a handling plan for thermally sensitive mutants selection. Part of it was reproduced on dishes in the presence of the agents mentioned above, with culture temperatures of 37 degrees for 24 hours, again using BCIP. When we used the green dye, we could select a first range of potentially interesting yeasts that have a lysis at 37 degrees. Then we replicated that on other dishes at 37 degrees C, preserving only the yeasts that don't grow 37 degrees, using the methodology with erythrosine B. And then the third selection criteria was growth at 37 degrees, so a limiting temperature on a YNB medium plus nystatin. And here we only kept the yeasts that were nystatin resistant and that would not grow beyond a temperature of 37 degrees. And now this is a summary of the three types of strains that we identified through the early screening tests. For Series A, phosphatase release at 37 degrees made it possible for us to select a certain number of strains which had a deep turquoise blue color. And then Series B, the colonies that were thermosensitive at 37 degrees on the dishes. We were interested in not seeing growth in colonies at 37 degrees C. And then Series C is a selection of mutants that would be assisted by the use of nistamisin. That made it possible to select a certain number of yeasts, and we went through further testing to validate the mutations that we had observed and come up with. So we went through more tests of oxotrophy and sensitivity at 37 degrees using the same methodologies as I've mentioned before. The strains selected were those that had gone through the oxotrophic tests and thermosensitivity tests. What's very important is that when you work with UV, ultraviolet, you have to also test the genetic stability in some tests, because some mutations can be reverted, and that causes you run the risk of losing any interest in the strain. The genetic stability was therefore tested on all mutants, and that would come up with a rather interesting number of mutants that could be relevant for creating protein medium for the yeast. Once we identified that, we took an interest in studying the cell content in terms of proteins contained in the cells. We used the Lowry method, which is pretty old. It dates back to 1951. And we used the SDS-Page electrophoretic methodology, which considers the band intensity in order to calculate protein content based on predefined markers. Here we have a certain number of proteins that were determined using the Lowry method. You always refer to a mother strain, and then you have Series A and B, from which we isolated interesting strains because they were better than the mother strain, which we could not use because it could not maintain the proteins of lysis at 37 degrees. This is the electrophoretic methodology. You see the bands of inactive proteins. Remember the electrophoresis that I showed at the very beginning, where we had the partial autolyzed material with a single band. Here you see, on the contrary, that you have different proteins with different molecular weights that are reactive, because when you have degraded proteins, you have some sort of smear on the electrophoretic band, which shows that with the what we've selected and through the analysis, we've come up with native proteins. Now, if you compare this optic reading, you can identify the presence of proteins for each strain of yeast. The outcome of this selection was that we started with 350 mutant strains that had been screened microbiologically, and all that work was done by Charpentier, who carried out that work with a certain number of students, because it takes a certain number of manipulations to come up with the selection of just 3 strains that were then screened for industrial scale production. So there's a lot of work to do for the selection of strains and the optimization of the process at a laboratory level. The industrialization stage is all but easy, because it's very nice to have interesting elements at a lab level, but before you can even propose a product based on yeast or protein extracts, you have to go through industrial engineering. So the strains that were selected for this industrialization were then submitted to the validation according to other production parameters. The first consisted in assessing the yield in terms of biomass production. It may sound stupid, but that has an immediate impact on the cost of the product at the source. If the production of biomass requires a long time or a lot of energy, that will end up having a direct impact on the initial cost of the product. And then you have to do the scientific work for screening all these products, and that has, which have to be translated into an industrial product that needs to be tested from an industrial and technical standpoint. Now, in terms of once the yields had been verified in tests of biomass produced, we had to see if we could use the native proteins at a high enough concentration. So we've had to optimize the recovery of the intracellular content without degrading the native protein content. And then the concentration, and then the drying up of the proteins, which is essential, because during the drying and concentration of proteins, you may see some degradation in some loss of quality in the product, which had to be evaluated. Once we started the pilot production at an industrial scale, we validated the functioning of the product. We tested that the product worked well. In the pilot stage of validation, we use the electrophoresis-based method, which makes it possible to immediately see that we have native proteins, because each of these columns, well identified, indicates the presence of native proteins. We have a certain wealth of native proteins with a molecular weight of more than 14 kilodaltons, which makes it possible to validate them for production from an industrial standpoint. We then went on to field test these products. We went through a certain number of tests on both reds and whites, and we realized that the doses to be used were between 5 and 10 grams per hectoliters for all red wines. Remember that the first generation product tests had doses of 20 to 30 grams per hectoliter, and not all red wines could afford to absorb that type of product efficiently. So the glue fining did not really, the fining did not happen effectively, and that had a bad impact on the amounts of polyphenols. So the dose of use now are in line with current winemaking practices. A very interesting thing is that the product works very well also with white wines. So even in the presence of very few polyphenols, such as in the case of white wines, there is an interaction with the yeast protein extract, because here the average dose is 1 to 2 grams per hectoliter. On some Italian wines, white wines, you can escalate that up to 5 grams per hectoliter because of the wealth of polyphenols that require the addition of these adjuvants. The idea is to replace for the standard products that are normally used for this particular type of fining. For instance, they consider egg albumin. In Bordeaux, egg albumin is still the standard, the golden standard for Bordeaux wines, whereas very noble glues in other productions are used, such as isinglass or fish glue, which we all know in terms of how well it fines and improves the product. In terms of concentration, the product does not require any adjuvants. Initially, the amount of polyphenols had to be rather high, but here, even without adding more tannin, the quality of the result is satisfactory. So the product in itself is self-supporting or self-sufficient, and gives you both qualities in terms of clarification or fining and the organoleptic improvement. These are more filled tests carried out comparing the protein extract and the egg albumin on turbidity. The dose of egg albumin was 25 grams per hectoliter, and it wasn't chosen 5 grams per hectoliter. It was not chosen by chance. It was designed to decide the optimum dose to have the optimum fining impact. That was 5 grams per hectoliter. So that was compared against 5, 10, and 15 grams per hectoliter of our product, and it was tested on 2 wines. The dark column is Syrah, whereas the light blue column is a Cabernet Sauvignon. In the fining with 10 grams per hectoliter, the level of fining performance is in line with the 5 grams per hectoliter of egg albumin. We then assessed the sensory impacts of this product. You have the control sample in green. Bitterness was eliminated in our samples, including albumin at 5 grams per hectoliter. Also, the control sample had drying tannins, which the other samples have to a lower extent. And there are more soft tannins, and so there is an improvement compared to egg albumin when it comes to astringency. That is very significant, and also the tannins are softer and less astringent. So all the tests that we have demonstrated that protein extracts from yeasts are very respectful of the wine quality in the fining process. Another impact that we obtained in light of the tests that were run on Cabernet Sauvignon, in this case, showed results that were similar. In green, you have again the control sample, and then in red, the egg albumin with the 3 extracts. You see that the volume and the astringency are largely improved with a significant reduction in the bitterness, which is systematically noticed with the use of EPLs, which are the yeast protein extracts. And also they work pretty well vis-à-vis the tannins. That is also observed on white wines. So this, in conclusion, is a product that provides a result which is similar to isinglass. It eliminates residual bitterness and makes it possible to improve the aromatic and profile and structure of the wine. This has to do with the tests run on red wine. So we have a test that was carried out on a white wine, a French white wine, a Muscadet, which was matured on the lees with a 4-month lees contact. We find the only this wine, the only glue fining agent for the wines before bottling is isinglass, and we've carried out various tests with the EPLs, and the dose is 1 gram per hectoliter, which is what is used, and it is equivalent to fish glue or isinglass, and you see the impact in terms of lingering or length on the palate, the bitterness, the volume, and so forth, and that is more or less the same result for isinglass as it is for the extract.
We believe in this product very much because it entails one advantage. It is a product which derives from an intrinsic wine product. It is the only product which is not exogenous to wine for fining purposes. This is something which is naturally present in wine. We concentrated it, we developed it in order to have results in terms of clarification and fining. Thank you very much. That was my presentation. Thank you.